Hello and welcome to Progressive Education Society's Modern College of Pharmacy, Nigri, Pune. In the subject Pharmacognosy and Phytopharmaceuticals for SY Pharm. Hello, myself Dr. Bhushan Pimple. Today in this video we will discuss about the morphology as well as microscopy of Cassia bark. Cassia bark are commonly known as dalchini in a grocery shop, and it is well known as Chinese cassia or also called as Chinese cinnamon. This differs a lot from cinnamon, which is Cinnamomum zeylanicum, which is a reddish brown color, thick and quill bark. But here you can find the blackish region or grayish region on the outer periphery. The biological source of this bark is these are dried stem barks of Cinnamomum cassia bloom, also called as Cinnamomum aromaticum because those are aromatic in odor, and Cinnamomum longifolium. Longifolium means the leaves are longer. These are the synonyms. This plant belongs to the same family of cinnamon, that is Lauraceae. Major chemical constituents are cinnamyl acetate, cinnamic acid, caryophyllene, coumarin, cinnamaldehyde, and styrene. The basically bark is used as a carminative or astringent. That means the oil, volatile oil obtained from the bark is majorly used as carminative or astringent. Besides, it is also used as a flavoring agent and stomachic. Stomachic means it stimulates the GI secretion and helps in digestion. It is galactagogue. It stimulates milk secretion in weaning ladies or mothers. And it is also used for the treatment of uterine hemorrhage, also called as postpartum hemorrhage. That means after delivery, the bleeding is stopped or it can be reduced by consumption of cinnamic acid or cinnamon cassia bark. About the morphology of this bark, its outer part is dark reddish brown, sometimes blackish brown in nature. Odor, it is aromatic, and taste, as far as taste is concerned, it is more towards the pungent side. That means it is more spicier and very slightly sweeter. The length is around 6 to 15 centimeter, but when the farmer cuts the bark and exports it, it can be around 40 centimeter as well, more than a feet. And usually what we get in the grocery shop is around 6 to 15 centimeter in length, 1 to 2 centimeter in diameter, or we can call it as width, and 1 to 3 in thickness. So it is too thin bark. Shape. The shape in which the bark appears can be channels. Channels are very similar to this U shape. Then quill, this is a quill shape. Then flat, the above surface is flat, and curved, slightly curved. So it can appear in these many shapes in the market. Special features are like, suppose these barks, they have small fractures, and inner surface it shows striation. If you zoom this, you will find linings over it due to the presence of many or numerous fibers. So these are longitudinal striations present on the inner surface. Now if you have to take this bark for microscopy, you need to cut it with a very sharp blade or new blade, and the bark is to be soaked in water for at least 2 days, or even you can boil it in water, but I will advise you to use the bark that has been soaked in water, cold water, for at least 2 to 3 days. Why? Because if you boil it in water, Since the bark has volatile oil, the volatile oil evaporates, and in the microscopy you won't be able to stain the volatile oil. So we normally prefer using the soaked bark or bark that has been soaked for 2 to 3 days in water. And while cutting, again, similar to the other practicals, here also the blade should pass in this direction. That means you will get the transverse section. Now, why we are interested in transverse section is because we are here taking the microscopy or studying the anatomy in order to see what are the different layers ranging from periphery towards the center. So we'll go to the microscopy part. Now this is the hand drawn sketch or a figure of Cassia bark. I have used this image because herein all the layers can be explained very properly. Later on we will be having a look at the section that has been taken in our practical, but this figure will help us cla clarify the various regions or tissues present in the TS. Like first, suppose periderm. Periderm is the outermost layer that is darkish grayish brown or dark brown in color. Second layer is cortex. Now this cortex, the major role of cortex is storage. Wherever cortex is present in the plant, it helps in storage of food material. So it usually stores the food material in the form of starch. Here you can find certain lignified cells, also lignified stone cells. These are the parenchyma cells that store the starch. Besides, they also have certain calcium oxalate crystals which are not visible in this figure. Now next layer is stone cell. Now, if you can remember, we have studied cinnamon in the earlier practical, and the cinnamon bark or the TS of cinnamon bark, it directly starts from stone cell layer. That is the reason why it is called as dried inner bark. And here, that means cinnamon lacks this outermost region. That means periderm and cortex is absent in cinnamon, whereas they are present in cassia. So I will not confuse you a lot by recalling all the again parameters of cinnamon. But here try to remember the first layer periderm, second cortex that is storage, third is stone cell layer that provides rigidity, toughness or protection. Then the fourth layer is the stone cell layer. Now the entire region behind the or below the stone cell layer is the secondary phloem region. This secondary phloem region has further distinct characters, like say suppose the medullary rays. These are the medullary rays which provide the nourishment in the lateral direction. Since we have taken the transverse section, these cells have been cut longitudinally. That means they are parallel to the direction of movement of our blade, whereas the other cells, what we can see here, are perpendicular to the direction of movement of our blade. So here we can see the longitudinal section of medullary rays. Next feature is acicular rapide, even that is not visible. Acicular means needle shaped calcium oxalate crystals. Oil cells. You can see here large oil ducts are present and they keep on secreting the aromatic oil. Then tannin. Most of the cells present in any of the bark, they do have tannin in them. Tannin is a protecting material. So this brownish spot, what you can observe here in this most of the cells, it is the tannin region or the cells containing tannin. Now this is the real image of cassia bar taken in our lab. So if I would have used this image first, it would not have been much more effective to explain the various regions. So you can see here perida, cortex, stone cell layer region, and the lower part is the secondary phloem comprising of medullary rays, acicular raphides. We will be having a look at these the images later on in detail, the tannin cells. Now, one by one, we'll have a look at the region. So, if you use Sudan red, you will find that the cork cells are stained pink in color. That means the outermost layer will show pink color if you use Sudan red here. In this section, it is not visible. Now, next is cortex region. In the cortex, you can see here there are a range of cells, and this cortical cells are slightly globular in shape, or you can say larger compared to cork cells. The major function of these cortex cells is storage. So they store the food in the form of starch. You can see here the starch. These are starch grains. On staining with iodine, they take up the iodine and they show purple or bluish color. Now, this is how, after staining with iodine, they appear. These are the bluish purple color starch cells, starch grains. These are numerous. A single cortical cell will have more than 8 to 10 starch cells. See here, this is the image that has been taken at 40x. All the images here are shown at 40x objective and 10x eyepiece. Uh, Here you can find in the cortex the acicular or sorry prismatic calcium oxalate crystal. Prismatic calcium oxalate crystal again provides rigidity to the bark. This is the prismatic calcium oxalate crystal. These are starch and these are starch stained with iodine. So far if you want to recall, we have used two stains. One was for cork, that is Sudan red, which gives pinkish color to the cork cell, and just below that is the cortex. In cortex, if you want to identify the calcium oxalate crystal, you can use acetic acid to clear all the debris. Here we have not used acetic acid, but to get the proper image of calcium oxalate crystal in any of the TS, you have to use acetic acid. And for staining of starch, you can use iodine. So these are the three stains. Next is the stone cell layer. Stone cell layer, as you can see here, this stone cell, these are dead cells and they are thickened from periphery. Outermost region is very, very thick and they do not have any cytoplasm in them. So this is the image that is without staining, and on staining with fluoroglucinol HCl, only the stone cell, they take up the, or they get converted to, since they are lignified, they show pinkish coloration. So this is the stone cell layer, and if you have noticed, you can find a beehive-like structure. जसं मधमाशांचं पोळं असतं, मधुमक्खी का छत्ता. Very similarly, you can find a bunch of cells in like a grape of bunch. The cells are arranged. So these are uh, pericyclic fibers. Pericyclic fibers and stone cell together, they help in providing rigidity. Very, they provide mechanical support, rigid mechanical support. That is the reason why the bark on cutting, it shows gives a crackling sound because of the rigidity. Part is the secondary phloem. This is the entire secondary phloem region. The secondary phloem region, as we have seen, shows the transparent large cells. These are called as oil cells, and you can find here the golden yellow color cells either in the group of two or individually present. These are called as phloem fibers. And other cells which are close enough and they have certain matter in them, they are called as phloem parenchyma. Now just have a look at this region. This is the area wherein you can find the longitudinally arranged cells just one below the other. That is in the they are stacked one below the other. These are the medullary rays. This is a medullary single medullary ray. This is oil cell. These are the phloem cells. And if you zoom in, as we have seen here, if you zoom in this area, you can find a secular, that is needle-shaped calcium oxalate crystal. Besides, they also show, this is what I call is phloem fiber, pericyclic fiber, sorry, and these are the phloem fibers. So this is the speciality of cassia bark. Now, once we have completed with cassia bark as well as cinnamon bark, many of you might be having the real confusion between the two. That is, what exactly are the differences? So here we will list the differences between the two, morphological differences as well as microscopical. First, we will focus upon the morphological differences. This is a cassia bark. It is obtained from Cinnamomum cassia and it is dried entire bark. Now this is an entire bark that is the reason why you can see outermost blackish region or grayish region. Then thickness, it is too thin. It appears in quill, channel or curve shape. This is a channel, this is a curve, and there is no quill here mentioned here, but we have seen in the earlier slides, it also appears as a quill. Color blackish brown externally, reddish brown internally. Now, if you compare it with the cinnamon bark is obtained from Cinnamomum zeylanicum, and it is thick, plus it appears to be quill, compound quill, double quill, as you can see here, double quill. Okay, then single quill, and the pieces also appear in channel and curve. It is light brown to reddish brown externally and dark brown internally. So these are the morphological differences. Atomical differences are concerned. This is a cassia bark. It has a presence of cork and the cortex. It is present. Fibers. The fibers here are short and thick. The stone cells, since we are observing them in the transverse section, you cannot find the length, but these are short. They have large starch granules and the medullary ray is uniseriate. Uniseriate means a single layer of cells is present in the medullary region. Here, although it is a diagram, this is wrongly drawn. Actually, it is a single layer. We have seen in the actual microscopic image. As far as cinnamon bark is concerned, the external, that is cork and cortex, these are absent in it. It directly starts from the stone cell, that is this region, stone cell layer region. It does have pericyclic fibers embedded into it. And if you look at the medullary rays, these are uniseriate or biseriate or triseriate. And the starch grains, these are, since they are mostly located in the cortex region and this lacks the cortex region, but yes, there are few starch grains in the secondary phloem. They are very, very thin or small, or sometimes even they may be absent even. One more difference between the two is the size of the volatile oil gland. Now, as far as cinnamon bark is concerned, it is highly aromatic compared to the cassia bark. It has large volatile oil and the diameter is more compared to the cassia bark. So this is the anatomy of cassia bark, dalchini, and this is the TS of cinnamon bark, also called as zeylanicum bark. So this is all about the differences between the two.